Hello students. In this class, I am going to discuss the different molecular techniques for industrial biotechnology. These molecular techniques are used for the separation and purification of biological products, and these are often used in the industry. In the previous classes, I have shown you some process, the slides. In that slides, I told you that industrial biotechnology is divided into two main processes, the upstream and downstream process. In these two process, mainly the downstream process is very much important because in this process, we are able to purify and we are able to get the various quality products, and these quality products are beneficial for human health and human society. So in the previous classes, I have told also this product isolation and purification is the is the main two stages that are used in the downstream processing. In this class, I am going to discuss about this purification process. Basically, the gel electrophoresis is one of the most used product purification technique by which we can generally separate the proteins and nucleic acids, and these are used in a huge manner that that the uh, purified proteins, that the proteins those are present in a clump may be purified and separated. So the basic principle of the gel electrophoresis is the run of macromolecules, biological macromolecules, through the gel matrices, and a electric field is applied here. And these macromolecules are separated on the basis of their charge and molecular weight in the electric field. The main important thing is that which is included in your syllabus is denaturing gradient gel electrophoresis, and in this class I am mainly focusing on this part. It was first used for the detection of base mutation in genomic DNA using the denaturing gradient gel electrophoresis, followed by the transfer and hybridization with the gel specific probes. And this was published in mutation research, and this type of fingerprinting was obtained due to the molecular separation of the DNA or nucleic acid on the gel matrices. The mainly this process is used to create the RDNA sequence databases, and simultaneously to create the phylogenetic tree. This DGG method is very much useful. For this process, we have to extract the environmental sample, and from the environmental samples, the DNA and RNAs are purified, and those purified products, if the product is less in quantity, these must be amplified using the PCR and RT-PCR, and after that the GGG is run. So this is the step which I am already discussed in the previous slide, that sample first purified, extracted from the environment, and the DNA, RNA must be extracted from the environmental sample, and they are now able to they are now subjected to DGG experimentation. So the principle lies in the DGG theory is the PCR amplified fragment must be the minimum length of the fragment is 500 base pair, but these are separate, and these are separated using a polyacrylamide gel with linear gradient of DNA gene naturants. Generally, formic acid and formamide and urea is used here, and the separation is based on the decreased electrophoretic mobility of the partially melted double stranded DNA, because there is a denaturing agent is present. The DNA double strand is partially melted on the basis of the denaturant concentration. Then these melting proceeds, the melting domain, the sequence variation within such a domain causes the melting temperature to differ. No one nucleotide difference can be enough here. The migration of the molecule is stopped at a particular position in the gel according to its melting temperature. And the attachment of a 30 to 50 base pair long GC sequence prevents the DNA from complete dissociation, which is known as the GC clamp, and it is very much important for the DGG profile. So in this slide, I have described how this process goes on. The denaturant intensity, the concentration is increasing towards the bottom line, and so that the DNA or the nucleic acid present at the bottom is more melted compared to the nucleic acid present at the upper region. The PCR products with GC clamps are applied on the top of the gel, and then the double stranded DNA fragments are moving in the gel due to the electrophorical power, and DNA the molecules made the increasing denaturing concentration, partially melting and decreasing the mobility. When these are separated, the molecular conformation is changed so that the matrix the movement through the matrix pore is hindered by its structure, and thus they are decreasing their mobility. And the DNA fragments are melted partially. GC clamp holds the strand together, and this is very important for the molecular binding pattern generation. In practice, this is the total experiment instrumentation which I am showing here. This is the polyacrylamide vertical gel electrophoresis unit, where the total DGG process can be taken place. Now the second thing is that it can be run in a horizontal gel also, where the denaturing agent is with the second dimension is placed in the second dimension in compared to the electrophoretic movement. It is the steep transition in mobility occurs at the denaturing concentration corresponding to the melting temperature of the lowest melting domains, and thus the different DNA sequence with same length can be separated due to this process. The temperature and duration of electrophoresis is optimized on the parallel gel, and thus this type of bending pattern can be obtained, by which we can easily separate the uh, same length DNA sequences, but their GC clamp, depending on the GC concentration, we can separate these DNA fragments. Now after that, the processing of the gel is done. The staining of the gel can be carried out with the iridium bromide and cyber gold solution, and the hybridization with specific oligonucleotide probe can be made after this. And and after the sequencing approaches can be done for the excision of, and this includes the excision of bands under UV light and reamplification of the fragments. And after that, the sequence of DNA fragments can be made for the phylogenetic analysis and probe designing. So this is the typical image shown here. In this, the dark colored bands are shown, which are represented the bending patterns, and these are useful for the construction of phylogenetic tree, and for the rDNA sequencing. And this is on the basis of some statistical analysis like UPMA and many other processes, the phylogenetic component tree can be constructed. The applications of DGG are described here. The study of community complexity can be studied with the help of DGG, and simultaneously the community change, that means any mutation, can be easily detected by the DGG technique. And also we can detect the micro heterogeneity of the rRNA encoding genes, and the comparison of different DNA extraction protocols uh, mm, also can be applied also can be made by the application of DGG techniques. It also used for the screening of the clone libraries, and for the determining PCR and cloning biases can be also determined by the DGG technique. There are some limitations also. This is the sample handling and extraction is much complex, and PCR is needed often. The electrophoretic techniques only display the rDNA fragments obtained from the predominant species, and no other DNA fragments can be we can observe in this process. The separation is relatively small fragments, limits the amount of sequence information. That means if the fragments are much larger, we cannot separate this type of fragments in this technique. It is not always possible to separate DNA fragments with a certain amount of sequence variation. The presence of multiple RRN operon with the sequence microheterogeneity and overestimation of the species richness can be done here. Beside the DGG condition, resolution of separation depends on the region of 16S RNA selected for the analysis. The co-migration of DNA fragments also happens here. So students, this is the class, and I think this will be helpful for your exam purpose. So please go through this slide, and if you have any question, you can ask me later. Thank you.